Welcome to another Diagnostic Test and Procedure class presentation. My name is Crystal Heron and I'll be your nurse practitioner student for this look at cultures and sensitivity testing. So now I get to go through the plan for this PowerPoint. What is on the agenda? First, we will start with a mini history lesson about microbiology. You probably barely remember your undergrad class, but I'll boil it down to a few fun pointers that you really don't need to know to take a culture, but I'll tell you anyway because I kind of like microbiology. You'll find that out. Next, I'm going to go briefly over the total extent of cultures and how I won't be presenting on absolutely every type. I picked a very general topic and needed to narrow it down just a bit. Then I will tell you about abscesses. That's going to be our beginning. I figured the best way to explain this type of testing was to start from one particular wound. So pus, good old pus. That is where our PowerPoint really gets on track on how to take a culture. Next, of course, I'll discuss the culture's journey as it goes to the lab and how it's processed in the lab. Then I'll get to where all you advanced practitioners really want to know, how do I help my patients? The last slides will include some information on antibiotics, final wound care notes, and just round out what happens after this results are in. So to start off our mini history lesson on microbiology, I'm going to introduce you to a few guys. You might know them as the fathers and founders of microbiology, and if you don't, you will after this slide. So let's start with Antony van Leeuwenhoek. He was a Dutch draper, later turned scientist. His hobby included lens grinding. In 1676, he pioneered the use of a simple single lens microscope, which he used to describe what he called, there's a Latin word for it, but in essence, he called tiny animals. These tiny animals would later be recognized as unicellular organisms. He was the first to document his microscope observations through his letters to the Royal Academy of Science. Some of his noted achievements that were documented on were he viewed muscle fibers, he viewed red blood cells, he was able to view bacteria, obviously, and even blood flow through capillaries. Next, we have Louis Pasteur. He is a French biologist, chemist, and microbiologist, best known for his work in microbial fermentation, pasteurization, and vaccines. He also, his research and work backed up and supported the germ theory. Next, we got Robert Koch. He was a German physician and microbiologist. He was big on techniques to isolate the different microbes. His most notable achievement was identifying and isolating Mycobacterium tuberculosis. In 1905, he won the award, the Nobel Prize Award in Physiology and Medicine for his research on tuberculosis. All right, Ferdinand Kohn. He was a German biologist. He is considered the founder of modern microbiology and bacteriology. He established the use of sterile culture mediums as a way to grow cultures that we use today. His work, he did work in classifications, and he proved that algae was a plant. He also created the 4 classifications of bacterial shapes that we use today: spherical, short rods, threads, spirals, and we use those classifications still today. All right, that wasn't so bad, was it? A little bit of history. Again, you don't really have to remember them. It's not important, but it's always fun to learn. So I had to put up some interesting pictures here, just a little break. If you talk to a microbiologist, sometimes their passion is just downright infectious. Pun intended. Online, this little trend popped up, a microbiologist making art out of microbes that they streaked on their plates. Thought it was very interesting. And then this one microbiologist, her son was playing outside in the dirt. He came in, she put his hand in a petri dish and grew what came off, and it's just a nice, beautiful depiction of the variety of microbes that live on us. All right, moving on to all the different cultures. So if there's a hole, we can culture it. If there's a liquid, you know we'll culture that too. And if it's a solid, you bet we can culture it. So you see, when I started this project, it was just one word, cultures. But I quickly realized that one presentation would not be enough to cover all the different ways that this general test can be used. So I had to get more specific, but I didn't really want to completely leave out all these other ways to collect microbes that are affecting our patients and how we culture them. So a little nod to everything. Blood cultures, anaerobic, aerobic, we use these collection bottles, and most of you have used them in the hospitals. If not, you know of them. Collection is done very cleanly. You actually prep the skin prior to using it to reduce the microbe population on the skin before you actually poke and draw the blood. It's also common to do fecal cultures. C. diff is in the hospitals. We test a lot for C. diff in the hospitals. Then we have our nasal swab cultures. Who doesn't have MRSA of the nares? I mean, really? But there's also many other different, we culture our environments to see how effective our cleaning products are. I work in the OR, and we have this guy that comes around with his little culture swabs. He does the surfaces to see how well they've been cleaned. It's culture a lot of things in this world. All right. Pus is the real star of the presentation, though. I narrowed this topic of culture and sensitivity testing by starting from what we learned in that I&D lab with those wonderful yogurt abscesses. So a patient presents to your clinic with a swollen, tender red mass on their chest. I'm going by this picture here. So mass on the chest, it's time for an I&D. You inject your local, you go get your sterile instruments, start setting up, and prepare to incise and drain this wound. So to culture, not to culture. Here's where a little research was interesting. So if a patient has no past medical history, they're otherwise healthy, and it's a small abscess with no signs of spreading infection, research shows you really don't need to do an antibiotic on this patient. You really don't have to do a culture and sensitivity on every single abscess. On this patient, the I&D should be enough. Pack it open, secondary closure, you're good. But if the patient has comorbidities, if there's evidence of increased spreading of infection, such as red streaking from the wound, a fever, increasing white blood cell count, then you might look at prescribing antibiotics and getting a culture for this patient. Wound infections often contain multiple different types of organisms, both anaerobic, aerobic. They can be gram-positive, gram-negative. Some of the most common pathogens isolated from wounds are Streptococcus, Staphylococcus aureus, E. coli, Proteus, Klebsiella, Pseudomonas, etc., etc. You get the picture. This is why identification can be so beneficial for our patients. All right. Brief review of I&D procedures. I have already seen somebody else's presentation on the I&D. I'm not going to go into a whole lot of detail, but I have to nod to the I&D because that's where we're going to get our culture. So once your consent's done, you go ahead and give your local, get your supplies set up, and you want to make sure you do a good skin prep. So the skin prep is important prior to getting your culture because you want to minimize and reduce the local microbes. You don't want to get the microbes that live naturally on our skin. You want to get what's in the wound. So good prep, drape it out, then you're going to incise it, wash it out, pack it open. Basic incision, drainage. Collection techniques. More is always best when it comes to samples going to the lab. So multiple thought process go into this collection. So first, you might take a syringe with a large needle and take a sample before you even open the abscess. This might afford you the less likely chance of contaminating the sample with the locals. But let's say that the abscess is already open, skin's compromised, and any low pressure expels some pus. You could put a syringe in there, try to get up, take out some of that pus for sample. You might even be able to expel that pus right into the sterile specimen container. But really, usually, the most practical, least invasive, and most effective method of collection is a culture swab, especially with patients where the abscess might have already lost most of its pus. In this case, you'll use a specimen swab to collect as much fluid as you can. Run the swab all along the inside edges. Make sure to go back and forth in a zigzag pattern. The goal is to collect as big a sample as you can. Use multiple swabs if you need it. Then you package it up in the test tube there, seal it, send it to the lab as soon as possible. All right, identification. Once the lab has the sample, the first thing they can do is prepare a slide for gram staining. A gram stain is a quick way for an initial report. Many types of samples can be examined this way. The material is stained to make the sample easier to see under a microscope. It is examined to determine the shape and the color of the bacteria that are causing the infection to give it a preliminary report. This can lead to some beginning help with the initial treatment plan for your patient. A little bit more on the gram stain. So think back to your semester in microbiology. It was a 4-step process that left your fingers purple. The method is used to differentiate 2 types of bacteria based on their structural differences of their cell walls. The bacteria that are able to retain the crystal violet dye are gram-positive bacteria, and then there are the gram-negative bacteria that don't retain that purple dye because their walls are thicker. The gram-negative bacteria are more resistant against antibodies because of these cell walls. Pair that with the shape, enables you to give you an initial narrowing of suspects prior to actually starting the culture. All right, to perform a culture, a part of the specimen is placed on a plate which contains a growth medium. This growth medium feeds whatever is growing there in the sample you sent. It is applied to the plate in a precise method. The idea is to get as many of these little tiny single colonies, these little round dots here. You just want the colonies themselves. That's where you get your sample from. You'll take, like this plate here has 2 different colony types on it. You'll take a sample from each one, put them in their own individual plate, and grow them on there. And that's how you isolate your colonies. Timetable needed for cultures, swab culture, basically 48 to 96 hours from culturing to sensitivities to the actual results. So Oh, agar plates. These are the fun little plates that we actually grow things on. So that previous plate was called a blood agar plate. So plates are petri dishes filled with a growth medium. Sometimes dye, sometimes specific growth mediums that allow for a selective amount of microbes to grow. So again, mixture of selective, non-selective plates are actually used. The blood agar was the first one. That one's mammal blood, usually sheep or horse. And that one is very non-selective medium, grows a lot of different bacteria, but it's also helpful to detect hemolytic activity. You have the basic nutrient-enriched plates. It's very good for growing all kinds of bacteria and fungi. Using different dyes and nutrients allows for the specialized plates to differentiate between these bacteria. And they're selective usually for what can grow. For example, we have a phenylethyl alcohol plate. It's selective for Staphylococcus, and it also inhibits the growth of gram-negative bacteria. There's the McConkey agar plate. That one was that bright pink plate, if you remember. It's an example of a specialized plate that has bile salts and crystal violet in it. It only allows the growth of gram-negative bacteria. Short and sweet of it is, by choosing several different types of plates, you can help narrow down what kind of microbe you're dealing with by what grows. All right, what you're really looking for in these reports. When you get your final report is going to be the identification of what's growing. It's also going to give you an estimated number of the microorganisms, and it's going to list off antibiotics and what microbes are sensitive or resistant to each. Last part is known as sensitivity, so that's what you're looking for. You want to know once. So what happens with the sensitivity report? Okay, so they streak these plates with the individual microbes. So they've already done their initial streaks. They've got very individual colonies. They grow those. So they take this plate, they streak it with just the individual microbe. They place these little bitty antibiotic discs all around. And the antibiotic discs are what you're checking for your sensitivity. So they place this in the incubator, they grow it, get it out a little time later, and this is what you see. So the area of no growth around the antibiotic disc shows what's susceptible. See, the larger the area, the more susceptible the microbe is for the antibiotic. So really, I mean, results that you're looking for. You're looking for an S, an R, or an I. So susceptible is that nice long ring, nice large ring around the antibiotic. You know those antibiotics aren't going to, or those bacteria aren't touching that thing with a 10-foot pole. Got a great sample there. You have ones that are resistant. That little pink disc there, that bacteria is laughing at it. It's like, what are you? But then you have ones that are intermediate. The kind of top right is pretty much a good example of that. And the idea is the intermediate ones are kind of your second choice. If, let's say the antibiotics that's susceptible to is what your patient's allergic to. So you might have to choose an intermediate one as a second choice. All right, antibiotics, broad versus narrow. After the IND, you're probably going to put your patient on a broad spectrum antibiotic because you want to get the infection under control as fast as possible. You've already cleaned it, you've packed it, you want to kill off anything that's causing troubles. But after the culture and sensitivity report comes back, you might just confirm that that is the best choice you've already made. But then maybe the antibiotic they're on, the bacteria are not so susceptible to, and you need to prescribe a different one. Patient allergies might also dictate whether that antibiotic is the best choice at knocking out the infection. Gold standard with antibiotics is to use the more narrow antibiotics whenever possible. That way we can keep the broad spectrum antibiotics from getting a lot of resistance, or a lot of bacteria getting resistance to them. Found this little graphic online. It was kind of fun. It's a great way to kind of show the example of the initial report versus when you get back what actually is growing. So the initial report is that top line there, gram-positive cocci, gram-negative, bacillin cocci. So you get that initial report within an hour of the lab getting the test. You're able to go down below and actually pick a antibiotic that covers both of them. So let's say it was gram-negative bacillin cocci in the sample. Ciprofloxacin covers both of them. Great starter. So just a fun little graph that shows how you kind of weed through or how you start things with antibiotics. But once that culture and sensitivity report comes back, hopefully you can narrow that down to something that's a little less broad, but it might not. That might be your best option. All right, you got your results for the culture, and that's it, right? Wrong. We are first and foremost nurses, and our holistic care of the whole patient doesn't stop at that prescription pad. So last slide is just a few closing pointers to remember for your patient. Education, education, education always is your best friend for prevention and further complications. Make sure you reiterate to your patients, finish the entire course of antibiotics, and don't just stop when it starts looking better. You don't need to save these for a rainy day, I promise. Send them home with wound care instructions. Is there packing? What's their need for a drain? When should they change out this bandaging? It sounds simple, but you really need to spell it out in black and white because once they leave your office, they probably won't remember it. Also, make sure they have a follow-up appointment, get with their PCP, send in the report, make sure they can get it rechecked so that they do get the drains pulled out, do get the packaging pulled out, and just make sure that there's not increased signs of infection. And send them home with a list of the signs and symptoms of things getting worse. Increased fever, new fever, increased pain, redness, swelling, if they have an abscess on the arm and suddenly they lose sensation in their fingers, things like that. You need to let them know when should they go back to you or when should they immediately go to an ER. Give them that list, make it simple, finish out the entire care of your patient. All right. Any comments or questions, please put them down in the comment section below. So until next time, this has been F&P student Crystal with your latest diagnostic test and procedure final class presentation. Thanks for watching.